Well, I'm going to tell you a nice story about how researchers can help companies to develop new products. One day I got this call from somebody saying, "You're a microbiologist, I think you can help us. We have this wonderful product that comes from hops that we think it might be antimicrobial and it's safe. The way we extract it is very safe, and we would like you to test it against all the pathogens you have in the lab." And I say, "Oh, this is a great idea." So that's what we did. We learned how to extract the antimicrobial products from the hops with a very safe procedure that does not involve any toxic, any chemical. And one day we found that this hop antimicrobial product was effective against a pathogen of bees. And they were really excited, and we really didn't know why, but the reason is that bees are in danger, and bees are very important for pollinizing crops. So we really want lots of bees around us for having good food. At the time that they contacted me, I had this wonderful postdoctoral fellow called Anna Motnenko that was looking for a project that could be related to developing a product for an industry. So I told Anna, "This is for you," and she said, "Yes, this is great, and I want to do it." So she worked really great at developing this product.
So we were just looking around and ran across a review article on honeybees. And the review article was talking about a pathogen that was affecting honeybees. And it was called American foul brood. And this thing had no cure. Basically, if you saw it in your hive, you were required by law to report it and then to destroy the bees and burn the hive. As we got into this a little bit further, we saw that there was a second bee pathogen that was also creating issues with the honeybees, and that was European foul brood. And lo and behold, it also was a gram-positive bacteria. And as we got further into this, we realized that we needed to do really a lot of bench work now that we have all of this data from this company. And that's when we reached out to Silvia, and we had this conversation. We sort of hit it off right away with the group and her lab and everything else. And with that, we created a lot of the data that that we would need for submission, as well as new data in terms of how can we formulate this extract to be able to go into hives. So that work continued, and in some places we started to find that we needed other expertise. That's when John sort of joined, because we needed analytical experience and an analytical lab. And we didn't have one.
I got contacted by my colleague Silvia Cardona in the Department of Microbiology, and she wanted to know if I would be willing to provide some expertise to help the company understand at the molecular level what they were observing with their plant extracts. We worked together, myself and the postdoc Anna, to do some of the chemical analysis to understand what the chemical identity and chemical profile of those bioactive molecules were that were present in those plants. The postdoc Anna that worked on this particular project, we wouldn't have been able to hire her to be in our laboratory space and help not only work on this project, but help train the other students in our laboratory. So it brings in highly qualified personnel into our laboratory space in a way that we wouldn't without participating in this collaborative project with this private industrial partner. This instrument is a nuclear magnetic resonance instrument, and it was instrumental for us to understand what the chemical structure was of the bioactive molecules that we were isolating from their plant extracts. So we could purify those molecules, and that strong magnetic field that exists at the center of this instrument allowed us to deduce the chemical structure of the bioactive molecules that were present in the plant extracts from Nature Recombined. And then behind me is something we call liquid chromatography coupled to a mass spec detector, or LCMS for short. This was an essential tool to allow us to characterize the bioactive molecules that were present in their plant extract.
We did, in fact, then start reaching out to other research groups, academic research groups in Western Canada. Because we needed honeybees, for instance, we needed to do field trials. And as it turns out, there has been a sudden increase in losses of honeybees. So the fact that the bees are dying is becoming an existential threat to humankind. Our mantra for our company is 4 words: no bees, no food. And so when we finally got to the point where we had enough data, we filed a US patent. And Silvia and John and Anna, they're a postdoc, they are also named on that as co-inventors. It was a collaboration and they deserve to be there.
In a much broader picture, we were able to contribute in, I think, a meaningful way to the development of novel and innovative biotechnology in a sector that I think is going to become increasingly important in Canada as we strive to kind of maintain our competitive advantage in some of these sectors. The agriculture sector, of course, is one of the biggest in Canada, and for me, I would have to say it was a real personal thrill to be able to contribute to novel innovation in that area.
This is what I really love, because you have a problem and then you work to solve it. And sometimes you get really successful and sometimes you don't. And so you have to sort of figure out how do you deal with that. But at the end of the day, like I say, the core for everything that I've done, all the pivots, has been the research. So, and that's what I love to do.